Good afternoon, everyone. I'm Patricia Davidson, Vice Chancellor of the University of Wollongong. Just while we're waiting for everyone to join us, maybe you'd like to share in the chat where you're joining us from today and any wishes that you would like to send Justin. So colleagues, we might get started and people are slowly joining us. As I mentioned, I'm Patricia Davidson. I have the honour and privilege of being the Vice Chancellor of the University of Wollongong in Australia, and it's wonderful to see colleagues from around the globe here today to celebrate Justin's exceptional achievements. It's such a pleasure to welcome each and every one of you here today. Today, we're here to acknowledge Professor Justin Yerbury, a celebrated molecular biologist, a friend and colleague to many, who is making significant contributions to better understand motor neuron disease. Before we start today, I would like to acknowledge country, and I know that people are joining us from places all around the world. And on behalf of the University of Wollongong, I would like to acknowledge that country for Aboriginal peoples is an interconnected set of ancient and sophisticated relationships. The University of Wollongong spreads across many interrelated Aboriginal countries that are bound by this sacred landscape, an intimate relationship with that landscape since creation. From Sydney to the Southern Highlands to the South Coast, from freshwater to bitter water to salt, from city to urban to rural. The University of Wollongong acknowledges the custodianship of the Aboriginal peoples of this place and space that has kept alive the relationship between all living things. The University of Wollongong acknowledges the devastating impact of colonisation on our campus's footprint and commit ourselves to truth-telling, healing and education. And we also acknowledge First Nations people from all around the world. We're so delighted today to be able to have this event to honour the work of Justin Yerbury and his colleagues. Justin was inspired to join the research effort to address motor neuron disease after discovering a family history of the condition and was already leading a research team when he himself was diagnosed in 2016. In spite of his own challenges and battles with this condition, he continues to lead his team at the University of Wollongong towards finding a cure. And beyond the four walls of the University of Wollongong, as we can see from people here today, he's motivated and inspired a generation of new scholars and scientists and spurred many all around the world to address this challenging condition. Today is an opportunity for us to be together with Justin's family, friends, and peers, and to acknowledge and celebrate his significant achievements and contributions to the University of Wollongong and to science. His achievements have been widely recognised. In 2020, Justin received an Order of Australia award, and in 2022 was awarded the keys to the City of Wollongong and the prestigious University of New South Wales Eureka Prize for Scientific Research. Justin joins us today, together with his wife Rachel, their daughters Talia and Maddie, and Justin's father Fred. A very warm welcome with you today, and thank you for sharing Justin with us. I encourage members of the audience to submit their own questions using the Q&A function, and we'll try to get through as many questions as possible. And now it's my great pleasure to introduce you to Dr. Luke McAlary, who will introduce you to the Yerbury Lab team here at the University of Wollongong. Welcome, Luke.
Thanks, Patricia, for the kind introduction for Justin and the group, and hello to everyone who's joined us today. So I am going to figure out how to share slides. Why is this not showing Oh, because PowerPoint's not up. Just apologies for a second. All right, there we go. Sorry about that. All right, so I've been given the sort of privilege to introduce Justin and who he is, and Patricia's touched on some of the things already that make Justin who he is, and this is sort of a bunch of slides about what I think makes Justin and such an amazing academic, which is why we're here to celebrate him. So Justin is a local, which is one of the most amazing things, because often you don't realize how many people in the local area are so amazing, especially scientists, and UOW has really helped us do that. But Patricia's already talked about Justin's situation in which he discovered that people in his family were succumbing to motor neuron disease, and he made the decision that very few people really do in their lifetime, and that is, I'm going to solve this almost unassailable problem myself. And that is exactly what he's been doing over the past decades, trying to solve one of the hardest problems that anyone or any really field of science could ever face. But I want to talk more about Justin, the academic. So what makes Justin an academic? And I split what makes someone an academic into three different things. So, and particularly for Justin, so what makes him an academic is that he is where's my point? Scientist, a beacon, and a leader. And I'm going to talk to these three things over the course of my presentation here. So first off, Justin as a scientist. So in science, we've got to publish research papers, and Justin's been extremely prolific at doing this. So since he started his career in 2005, he has published over 64 academic research articles and has been cited over 5,500 times, which is no easy feat. He is, in collaboration with other researchers around the world and Australia, obtained over $16.8 million in competitive funding since 2007, which again is a major achievement and not really something that a lot of people have achieved. As Trish said, he was the 2022 Eureka Prize winner, which is a very prestigious Australian science award. He won the 2022 Premier's Prize for Medical Biological Science, another very prestigious award, and was a 2020 Order of Australia recipient, which is a huge sort of Australia-wide recognition of what Justin has done. So what more about Justin's science? So I'm not going to go into too much detail, but there's two major areas that I think Justin has really, really helped in the MND field. And area number one is this concept of supersaturation of proteins in motor neuron disease, of which Justin was one of the key driver in this research. And that's been a real big impact on the field and how we understand the disease as it stands. But you're going to hear more about this from the lab research team. And then also Justin's contribution to the idea of prion-like propagation in MND, which is another important way of understanding how this disease occurs, which again, you'll hear from the rest of the research team. So the second thing I'd like to talk to is Justin as a beacon. So Justin has had global collaborations with leading scientists around the world involved in motor neuron disease and in general, an idea called protein homeostasis. And Justin himself is a key figure in the Australian MND research community, often being found talking to pretty much anyone within that community who is serious about curing this disease. Justin knows pretty much all of them, which is excellent. And he's also a supporter of open science and shares pretty much anything that he has, whether that be ideas, reagents, and knowledge. And so I want to show off just how much of a beacon he is by showing you sort of a small snapshot of his global collaboration network. So here's researcher Neil Cashman from Canada, Jacob Ayres from the United States, Giorgio Favren from Cambridge, UK, Justin Benesh from Oxford, Chris Dobson, one of the major probably collaborators Justin had and a dear friend of Justin's, Michele Vendruscolo, Janet Comita, Benedetta Bolognese, Stephen Oliver. And yeah, so you can see there's a big concentration around Cambridge, and that's because Justin spent some years there studying with Chris Dobson who was one of Justin's close friends. And so I'd like to narrow down now on Australia, because there's a few of these people here and it's quite a big network in Australia. So you've got all these people from all across Australia who have collaborated and worked with Justin, many of them quite closely, and have really contributed to Justin's science, and he has contributed to their science as well, because we don't do research in a vacuum. Often it's a team effort, and this is a team of people that Justin was able to create. And so speaking of teams, the local UOW team is especially important because there wasn't any M&D research going on at his university until Justin started, and he managed to get local experts such as Heath Eckroyd, Lizanne Uy, Mark Wilson, Cara Perro, and Ron Sluter, some of whom you'll hear from later in the session today, to really start engaging in this research here. And Justin was a big part of that. And so the final thing I want to touch on for who Justin Yerbury is as an academic is a leader. So he's been leading a research team since 2007, and he's had 21 students graduate, 10 of those PhDs, 11 of them honors, which is, again, a hard thing to do, to mentor someone through such a trying time in their life. And he also has the best looking team at UOW, I believe. And so just to show you how good looking they are, I've made this slide here. And this slide is sort of like, it's a montage I made, which took quite a while. But these are the people, past and present, who have worked closely in Justin Yerbury's lab. And some of these people still work here with Justin, and some of them have gone on to great things. Some of them have gone on to become postdoctoral researchers, scientists in their own right, or some of them have gone on to work in industrial capacity or have left science altogether. But not a person in here can say that they haven't had some part of their life affected by Justin's drive, passion, and love for research. Yeah, where are those last ones popping up? There they are. All right. And so this is really the team of people that Justin has built up over the years. And with that, I'd like to bring it back to the three major things that I think make Justin the academic: a scientist, a beacon, and a leader. And I want to reiterate that again. Justin Yerbury, the scientist, the beacon, the leader, and the absolute legend. And so with that, I will close my remarks and I'll open it to Natalie Farrell, who has worked with the Justin Yerbury for the last 10 years and is sort of the center of the laboratory, really. She is an expert in her own right. So I will begin playing that. So let me stop my video.
Hi, my name is Natalie Farrell, and I'm a senior research assistant that's had the pleasure of working with Justin for the past 10 years. Today, I'm going to give you some brief background on MND and Justin's research and highlight some of the models we use to study the disease in the lab. So motor neuron disease, or MND, is a fatal neurodegenerative disease characterized by the death of motor neurons, or nerve cells. These nerve cells control our muscles responsible for voluntary movement. And as MND progresses and the motor neurons start to die, these muscles start to waste away, leading to paralysis and eventually death by respiratory failure. The average life expectancy for someone suffering from the disease is around 2.5 years. And while each day 2 people die from MND, another 2 are diagnosed with the disease. The cause of most cases of MND are unknown. And importantly, currently there are no effective treatments for the disease. The pathology of MND is marked by the deposition of large protein clumps called protein aggregates within motor neurons. Now, the proteins within these aggregates vary depending on the genetics of the disease. And here are just a few examples of some of the proteins you might find within these aggregates that are associated with the disease. So SOD1 was the first protein identified to be associated with MND, and a lot of today's talks will focus on this protein. Now, it is not yet known whether these protein aggregates are a cause or consequence of the disease, but a major focus of Justin's research has been how these aggregates disrupt essential cellular processes, which ultimately results in the death of motor neurons. So let me tell you a little bit more about the process of protein aggregation. In a healthy motor neuron, we have millions of proteins moving around our cells. And these proteins carry out a variety of different functions, such as moving molecules from one part of the cell to another. In order to perform their function, proteins need to be folded into their correct shape or form. If we liken these proteins in a cell to Maltesers in a flask, you can see that the proteins, or the Maltesers in this case, are able to freely move around the flask. However, under conditions of stress, such as a mutation in a gene associated with MND, these proteins become misfolded and sticky, making them prone to form these protein aggregates that we see in the motor neurons. If we go back to our Malteser analogy and expose our Maltesers to a bit of stress, such as heat stress, you can see they become sticky and form clumps that are no longer able to move around the flask like they could before. And this is what we see happen to the proteins in motor neuron disease. There are a number of ways we can study the disease in the lab, and more often than not, our first port of call is a cell culture model. So we can culture motor neuron-like cells in a dish and introduce MND-associated proteins such as SOD1, which can be tagged with a fluorescent protein, such as the green fluorescent protein from a jellyfish, which allows us to visualize their distribution within the cell. And we can use these models to assess the effectiveness of different treatments, such as a drug treatment. So you can see in this case, we've got some cells that contain our SOD1 protein, and without any drug treatment, we see the formation of these large protein clumps or aggregates within the cell. However, when we expose the cell to some drugs, there are no longer any visible aggregates within the cell. So once we assess the effectiveness of treatments in our cell models, we can move on to a preclinical model of the disease. Now, there are a number of preclinical models out there that recapitulate MND pathology. And together with our cell models, they give us a better understanding and a bigger picture of what's happening in the disease. So now I'll hand over to my colleagues who will go into a bit more detail about the work we are doing with some of these models and how they help us understand what's going on in MND. Thank you for your attention. Hi, my name is Natalie Farwell.
Hi, my name is Jeremy Lum, and I've been a postdoc in Justin's lab for the past 3 years. Working on to investigate new therapeutic options to prevent the protein aggregation that Natalie was talking about. So as Natalie said, that within motor neurons, there's hundreds of millions of proteins in the cell constantly being produced. And the one protein that we're particularly interested in is this SOD1 protein. And when it's produced and newly synthesized, it kind of comes out a bit like a blank piece of paper, like Natalie said, and it requires intricate folds like origami to produce the final folded functional form. However, when in MND, when there's a mutation in this SOD1 gene, what happens is the protein is more likely to misfold, and that's an intermediate to form these big, large aggregates. And it's the accumulation of those aggregates that we believe causes motor neurons to start to die. So of late, in the ALS field, there's been this drug called copper ATSM, which has garnered quite a bit of interest. Now, the reason for this is it's thought to stabilize the misfolded SOD1 protein and prevent it from aggregating. So some work from Natalie and Madison Yerbury, Justin's daughter, used the motor neuron-like cells that Natalie mentioned before that express our SOD1 protein, and you can see that it forms those aggregates. However, when they put copper ATSM onto these cells, they found that the aggregates were no longer visible. And when we quantify that, we can see that without copper ATSM treatment, we see a high percentage of cells expressing aggregate proteins. However, when treated with copper ATSM, it drastically reduces. So following on from that, what we did was we moved into a SOD1 mouse model of MND. Now, this mouse model has a human SOD1 gene in it with a mutation, similar to what we observe in the clinic. And as these mice grow older, those SOD1 starts to misfold and produce those aggregates similar to what we see in the dish, and they develop paralysis as well. So they become a model for us to test drugs in. And what we did was we treated these mice with copper ATSM. And you can see here that copper ATSM increases survival of the mice that don't receive any treatment at all. Now, it's studies like this that have led to copper ATSM actually being investigated in human clinical trials currently. So Justin kind of went back to this, how does copper ATSM work? And based on the fact that it's now in clinical trials, he thought, well, you know, it must be doing something good, hopefully. And he went back through and started to search for other compounds which may be able to stabilize that misfolded SOD1 protein. So some work from Dr. Luke McCallery and Victoria Shepherd in our lab found two other compounds which can help stabilize this misfolded SOD1 protein. And they're called telbivadine and epsilon. Now, the best two things about these drugs is the fact that they're already being used in the clinic in the case of telbivadine, and epsilon's already in clinical trials. So that drastically reduces the amount of time, if successful, for these labs to be tested in safety trials. We already know that we can deliver them to humans without too much toxic side effects. So what we did is we went back to our cell model and we put the three drugs on the cells to see whether they reduce aggregation. And you can see here that copper ATSM, telbivadine, and epsilon all reduce the amount of cells with aggregates compared to those that are untreated. Secondly, we looked at cell survival to see whether the aggregation load was also correlated with reducing that motor neuron death. And we see that copper, ATSM, telbivadine, and epsilon all increase cell survival as well. However, then Justin had this other idea. What happens if we add all three drugs on together? And we call this combination therapy. So when we combine all three drugs, we see that we reduce the amount of cells with aggregates even further. Similarly, when we look at their cell survival, we see that we increase the cell survival more than any one drug alone. So this was quite promising. We then went back to our SOD1 mouse model of MND, and this work was done with Michaela Brown. And we see that we treated the mice with either nothing, copper ATSM alone, or copper ATSM, epsilon, and telbivadine being our combination therapy. And the first thing we wanted to do was test whether these mice had improved motor function compared to the untreated mice. So what we do is we put the mice on this apparatus called a rotor rod, and this rod spins around and around. And as disease progresses and the mice develop paralysis, they're unable to stay on the rod for as long. And what we do is we measure the time to fall off. So you can see here that the untreated mice aren't able to stay on that rod for very long. However, when we treat with copper ATSM, we can increase the time that they stay on that rod. Furthermore, when we treat with our combination therapy, you can see they're able to stay on the rod even more. And lastly, we looked at survival of these mice. And as I showed you before, copper ATSM increases survival compared to those untreated mice. But promisingly, when we add our combination therapy to these mice, we can extend their survival more than copper ATSM alone and the untreated mice. So this is promising work from the lab. And it kind of suggests that if we target this protein aggregation process, we can find potential therapeutics for MND. And lastly, I'd just like to show this video from the 2014 Ice Bucket Challenge that we held here at UOW. And I want to show this video because I think it's quite important, the fact that the Illawarra community has donated a lot of money to MND research, and in particular, Justin's research. And on behalf of Justin and the team, we really want to say thank you to the Illawarra community for those donations, because a lot of the work that I've shown today was actually funded through those donations. And now I'd like to pass on to Kristen Chisholm, who will be talking about how to remove these protein aggregates from the cell.
Hi everyone, welcome. My name is Kristen Chisholm. I'm a PhD student and a research assistant for Justin. And as Jeremy said, today I'll take you through my work, which has been to develop a genetic therapy that targets misfolded SOD1. So as we've already made clear, one of the main research areas that we focus on in the Yerbury group is how to protect the cell from misfolded protein. We know that if there's too much misfolded protein, it accumulates into these insoluble clumps that we call aggregates, which end up in the death of the cell. Jeremy talked about using some small molecules to help proteins fold correctly. But for my project, Justin had a different question. He wanted to know whether we could actually remove the misfolded protein and in that way prevent the aggregates from forming. So in order to answer this question, we had two problems that we had to face. The first was, how were we going to find the protein that we were targeting? You've got something like 42 million protein molecules in each one of your cells. So finding the right one is no mean feat. And then secondly, if we could find it, we then needed to know, well, how are we going to remove it? So to answer the first part of this question, Justin formed a collaboration with a Canadian biotech company called Promise Neurosciences, and they had developed this panel of antibodies. Now, antibodies are quite a useful tool for biologists. They are highly specific. So these antibodies could not just find SOD1 in the cell, they could actually differentiate between the misfolded form and the correctly folded form, which is really important for us because we want to leave the correctly folded form alone so it can do its job. So for the second part of the problem, we turn to one of the natural pathways in the cell. Now, as we sit here today, it's not just the people with MND who have a misfolded protein problem. We all have misfolded proteins in our cells. Some estimates suggest that up to 30% of proteins misfold. So it's quite a complicated process. But luckily for us, the cell has a mechanism to protect itself. You have these surveillance proteins called E3 ligases, which travel around the cell looking for misfolded protein. And when they find one, they bind to it, they tag it, and then they transport it off to this structure here. We call it a proteasome, and it's a little bit like a garbage chute. You feed the protein in the top, and it gets chopped up inside into these small pieces that are harmless to the cell. So Justin's strategy was quite straightforward. Let's take the antibodies from Promise, let's fuse them to an E3 ligase, and when we put them in the cell, the antibodies will find misfolded SOD, and the ligase will then take it off to the proteasome for degradation. So we call our therapy misfoldable because it is specific for misfolded protein. In my case, it's specific for SOD1. And the ubiquitin ligase pathway, the ubiquitin being the tagging molecules and the ligase being the surveillance protein. So over the past 3 years, I've been optimizing this therapy. I started by in a cell culture model. Promise sent us a number of different antibodies, and I also trialed a number of different ligases. And you can see this is just a representative graph of the kind of results that I was finding, where without the misfoldable, you can see that somewhere in the vicinity of 50% of my cells were showing aggregates. But when I put in the various misfoldables, I was able to reduce the number of cells with aggregates. Now, with that success, we decided to take our misfoldable into a mouse model, which Jeremy spoke about. So right now we're hoping that the mice who have both the SOD1 mutant gene and my misfoldable are able to retain their motor function for longer and live longer than the mice that just have the SOD1 mutant protein. So with that, I'd like to thank Fight MND, who have funded my work, and the scientists who've helped me along the way. And just before I go today, I do have something personal that I wanted to say. I first met Justin in 1991. He was, that was long before SOD was found, or we knew that motor neuron disease affected families, that some people had a genetic reason for it. He had just finished high school and he was going off to uni to study commerce and be a professional basketballer. And he met my friend Rachel, and not long after that married her and had his 2 beautiful girls. Now, over the next 25 years, I watched his journey from understanding that his family were affected by this disease that we knew very little about, to trying to understand it by going back to uni to study science, to becoming this world-renowned leader in the field. And in all that time, I was blown away by his dedication, his commitment, his resilience, and his intelligence. But it wasn't until 2018 when he chose to be ventilated in order to continue his work fighting this dreadful disease that I realized how deep his strength really was. At the time, I was leading a fairly comfortable life as a high school science teacher, but I was so inspired to be a part of his fight that I decided to come back. To Justin today, I want to say to you that we see your superhuman strength. You have inspired all of us. You inspire all of us every day to work with you. And I know that technically you're my boss at the moment, but as your friend, I want to tell you how proud I am of everything you have achieved and how honored I am to be doing this work with you. And with that, I'm going to pass on to Dr. Isabella Lambert-Smith.
Thank you, Kristen. Hi, everyone. So my name is Isabella, and I'm going to be outlining a particularly ambitious and novel treatment idea that Justin came up with a couple of years ago. A strategy that we hope will lead to a regenerative therapy for people with MND to restore their muscle function. So I'd like to start by highlighting that Justin's research vision is continually evolving. Natalie, Jeremy, and Kristen have all outlined this process, this concept of protein misfolding and the pivotal role that this phenomenon has in MND. So understanding this process of protein misfolding and discovering ways to correct it has formed a core part of Justin's research vision. But so yeah, in order to be able to save motor neurons and treat MND. However, regardless of finding ways to stop protein misfolding or find ways to clear these toxic proteins away from motor neurons in order to save the motor neurons, up to 80% of a person's motor neurons have already degenerated by the time that they are diagnosed. So Justin realized that in addition to finding ways to save motor neurons that still exist in a person, we also need to find ways to replace the motor neurons that have degenerated so that a person with MND is able to regain their muscle function. So as Justin's research vision expanded into the realm of regenerative therapy for MND, he came up with this idea of investigating how we can actually replace motor neurons that have degenerated. And he came up with this idea of using the body's own healthy cells to regenerate motor neurons. So this is a vital research direction because even if we do find ways to save remaining motor neurons in the person and stop the progression of the disease, a person won't be able to regain their muscle function without additionally receiving regenerative therapy to restore the connections with muscles that have already been lost. So Justin came up with the idea of investigating if we can use sensory neurons, which are unaffected in MND, to convert into motor neurons. So this might sound like science fiction, but this process of cell transdifferentiation, which means converting one cell type directly into another cell type, is an area of research that has arisen in recent years. So this forced conversion can be achieved using a combination of chemical cocktails and by modifying the activity of certain key genes. So you might be asking the question, why would we target sensory neurons for transdifferentiation into motor neurons? Won't a person lose their sensory function, and is this worth it? So motor neurons are a very unique type of cell in the body. There are actually no other cell types that are quite like motor neurons. They are extremely long cells. So they connect our spinal cord to all of the different muscles in our body, and thus they can actually span for up to 1 meter in length. There are no other cell types in the body that are this long and cover such a large distance, except for sensory neurons. So sensory neurons are similar to motor neurons in that they can actually span up to 1.5 meters in length, and they can lie close to motor neurons in the body, connecting with muscle. So a combination of Justin's ingenuity and his strong network of collaborators has allowed us to make use of 3 very powerful scientific model systems to assess our strategy for motor neuron regeneration. And so this includes strong ties with Professor Lizanne Uy, who we will be hearing from a bit later today, and Dr. Yi-Lian Chu, who is a pioneer in her field using the nematode model C. elegans. And so just to summarize all of this, Justin believes that we need to find ways to replace motor neurons in addition to using conventional therapies to save remaining motor neurons in people. And this is important so that a person with MND will be able to both have their progression of disease slowed down, in addition to actually regaining their ability to control and move their muscles. So Justin has been truly a pioneer in the field of MND research. He has brought together scientists from literally around the world, coming from different scientific specialties, coming together to work on MND and to find effective ways to treat it. And he's also brought in the minds of scientists who work on other diseases. And this includes his friend and fellow scientist, Darren Saunders. Justin, throughout his career, he has challenged traditional perspectives in the MND research field and has significantly advanced our understanding of MND. And to reiterate what Luke said earlier, we all see Justin as a leader and a beacon. He has brought together and trained up so many different scientists to work on MND. And his family's journey with MND and his journey to becoming a leader in MND research, his kindness and his resilience and his generosity to all of us that he has mentored in his team are truly remarkable. And I think it's safe to say that our lives and our perspectives have been changed for the better because of him. So thank you, Justin. So I will now pass on to Senior Professor Mark Wilson. Thank you.
Hello, my name is Mark Wilson, and I'm just going to give a brief talk today on one of our major projects, discovering new drugs to treat motor neuron disease, and we're searching amongst Australia's unique flora and fauna. And this project was originally inspired by an ex-PhD student who's shown here second from the right. So that's Justin, around about 2005. And 2 other students, Amy White on the left and Elise Stewart on the far right. And that's me looking somewhat bemused on the second from the left. So Justin was quite an outstanding student, both committed and talented, which is a rare combination. And here he is at his graduation wearing a funny hat in 2007, and his PhD thesis, characterization of novel extracellular molecular chaperones and their effects on amyloid formation, gave him the basic grounding in studies of protein folding and chaperones, which led him to then go on to his focus, which has stayed with him, of motor neuron disease, which involves protein aggregation. So some of the other talks may have introduced this already, but in motor neuron disease, insoluble lumps or inclusions of protein appear inside the nerve cells that are joined to muscles. And when these inclusions, shown now by the bright green circles inside the nerve cells appear, it leads to the eventual death of the nerve cell and consequently a loss of control of muscles. Okay, so yes, nerve cells die, you can't control your muscles anymore, pretty serious pathology. So we use a cell model which is transfected, meaning it has a gene introduced to express a particular protein that is found commonly in motor neuron disease, and that particular protein is called TDP43. And TDP43, we fuse it, in other words, connect it end to end with a fluorescent protein, and we use this and express it in cells. And the little video here shows a cell rotating in three-dimensional space, and the bright green lumps are the inclusions inside the cells. So we can make cells express this protein and they form the inclusions, and this has bad effects on the cells themselves. So we use that model as a way of searching for compounds that would reduce those inclusions and lessen the pathology associated with motor neuron disease. So we grow those cells in 96 well plates, plastic plates, tiny little wells, and into each well we add different drugs or extracts from plants or animals. And then we grow the cells for about 2 days, and then we break the cells open. So we then analyze how many inclusions have been formed inside the cells. And to do this, we use a technique called flow cytometry. And basically you burst open the cells with a detergent, and then you put the lysates, the solutions with the disrupted cells, through a flow cytometer. And we are searching for, like this dog is looking for a particular type of drug, we're looking for drugs that reduce the number of inclusions formed in the cells. And an unusual feature about this particular project is that we access fractions, chemical fractions, that have been prepared from natural Australian native plants and animals. And there's a thing called Nature Bank, which is up at Griffith University in Queensland, that provides all of these, they're like libraries of extracts from different types of plants and animals. And so we screen literally tens of thousands of these extracts, looking for those that might reduce the number of the inclusions in the cell model. And this is just some data showing a plot on the left top here, and the red circle represents 3 fractions that were screened that have substantially reduced the number of inclusions. So the percentage of inclusions of the control is shown on the y-axis at the left. So the other fractions to the right that are not encircled with a red circle are just a variety of other fractions that do not reduce the number of inclusions. And so they're running at around about at the 100% level, whereas the 3 inside the red circle are down at more like sort of 30 to 40%. So quite a lot of reduction. We then took one of those fractions in the red circle and had it chemically further separated and then retested it. And the plot on the right shows different fractions from that secondary separation, and then they're tested in the same assay in the same way, and you can see that fractions 54 and 55, these ones down here, show a very marked and dose-dependent reduction in the inclusion numbers. So, and this particular fraction comes from a marine sponge. And so we're continuing work on this to try and purify and get the specific compound from that marine sponge that has this activity. So that's where the arrow shows where you have the decrease in numbers of inclusions. So this project is continuing, and in this year, 2023, we have new funding for it from both MND Research Australia and also from Takeda Pharmaceuticals. And we are continuing this search and hope, with luck, to find a pure compound, or maybe more than one, that perhaps can be translated into an effective treatment for motor neuron disease. And so, to go back to the beginning, this was inspired by Justin himself. Thank you.
Thanks, Mark, for that presentation. I'm Professor Ron Sluter, and I'm
delighted to talk about our work with Justin on motor neuron disease and to also take this opportunity to publicly congratulate him on a magnificent career to date, including the publication of his forthcoming book. If memory serves me right, I first met Justin in spring of 2009, and shortly after he came into my laboratory with some excitement, showing me this article on motor neuron disease and on purinergic signaling, which was the focus of my research. This article was from Cinzia Ferlante in Italy, a name that I'll come back to later. This article, as well as other articles at the time, highlighted the fact that if you activate P2X7 on brain cells such as microglia and astrocytes from mice with motor neuron disease, this would cause the release of unknown factors, which in turn would induce toxicity in motor neurons, which then has the potential to drive MMD progression. As a result of this study and others at the time, Justin and I decided that we would look at this pathway together in motor neuron disease. And so we recruited a talented research student, Rachel Bartlett, who's now Dr. Bartlett, and as you've seen from earlier presentations, is still in Justin's lab working. As part of Rachel's early research, she confirmed that microglia cells express P2X7 and that its activation can cause the release of reactive oxygen species, which may be responsible for inducing motor neuron death. This research that we did previously with Justin is really just an example of his incredible ability to work with UOW researchers to raise awareness around motor neuron disease and to really develop a critical mass of MMD researchers here at Wollongong, which Luke highlighted earlier. In addition, Luke also highlighted the fact that Justin's had many national and international collaborations, one of which is with Professor Neil Cashman from the University of British Columbia. If memory serves me right, I believe Justin tracked down Neil Cashman at an airport after a conference one year to discuss this idea of the prion-like propagation of protein aggregates going from one motor neuron to the next. And so together they've done a number of years' work on that subject, and Luke alluded to that previously. What wasn't known at the time was how these protein aggregates could be released from motor neurons. So Rachel Bartlett in our group continued to look at P2X7 on motor neurons. She confirmed their expression on these cells. And then if these cells express protein aggregates and you activated P2X7, you could get release of these aggregates. More importantly, Rachel showed that these aggregates could be transferred to nearby motor neurons where they would induce cell death and promote motor neuron disease progression potentially. In addition, these aggregates could also be taken up by neighbouring microglia to cause the release of inflammatory mediators, which also have the potential to drive motor neuron disease. With Rachel Bartlett, we then went on to undertake the first preclinical drug trial of MMD in mice at UAW, and this work laid the platform for other studies that Justin's teams talked about earlier today. In this study, Rachel treated mice with a non-selective P2X7 agonist a P2X7 antagonist and could reduce motor neuron disease. At the time of our study being completed, 2 other studies were published supporting this idea that if you block P2X7 with drugs, you can prevent motor neuron disease in mice. This then led us to trying a second P2X7 drug, one that was more selective and more potent. And this work was conducted by Dr. Diane Lai, a UAW graduate, and at the time a research fellow in our labs to undertake this study. This study that Di completed along with another one that was published from a different group collectively showed again that if you target P2X7, you can reduce motor neuron disease in mice. However, it highlighted the importance of dosage and starting points of treatment. And so just in conclusion, the work that we've done with Justin over the years has been internationally recognised, and we've recently completed a book chapter with Cinzia Ferlante, who opened the fields of P2X7 to motor neuron disease and motor neuron disease to P2X7. And this chapter, as well as future work, will continue to explore the role of P2X7 purinergic signaling in motor neuron disease and hopefully contribute to finding a cure for this disease. So thank you for your time, and I'd now like to introduce my colleague, Professor Lisanne Uy.
Hi, everyone. It's my absolute pleasure to be able to talk to you today. So I'd like to tell you a little bit about some of the work that we've been doing on motor neuron disease as a result of Justin's influence. So I first came to, progressing. There we go. I first started my lab in Wollongong in June 2012, and it was shortly after that I gave a talk at one of the centres and talked about the work that we've been doing using patient cells to understand Alzheimer's disease. And Justin and Mark came to me and asked if we could think about doing a similar thing for motor neuron disease. So we started to do that, and we wrote a grant together. So myself, Justin, Ian Blair and Dominic Rowe, who are from Macquarie, and we received our first funding from the MND RIA in 2014 to progress that work. And we started to build a library of patient cells from MND patients from the MND clinic in Macquarie at that time. So that really kicked off a very fruitful, productive and fun collaboration with Justin and his team and other collaborators from around Australia and in the end internationally as well. So I'm showing you some pictures here that go through a little bit of the history of that collaboration. And one of those pictures is of Justin and I in 2015 after we'd received a big grant with some collaborators at Macquarie and other universities as well. I also show here a picture from 2014, which is from the proteostasis and disease conferences. So we had this, and we still have this very vibrant research community focused on proteostasis and including motor neuron disease as well. And that picture shows you a picture of Ulrich Hartl, who's one of the world's foremost leaders in protein chaperones. And it just goes to show that we've been able to bring these leaders to Wollongong as part of the work that we've been doing. And this amazing community that we have for research is also shown here from a picture in 2017 where we were celebrating MND research at UOW. So a little bit more about my lab. So we are the Neurodevelopment and Neurodegeneration Lab. So I'm showing you a picture here from 2022. Actually, that was from the proteostasis meeting last year. So that was the 4th proteostasis meeting that we've had in Wollongong. But I also wanted to show you this picture from 2014. So in 2014, just when Justin and I started the project where we wanted to use the cells from patients to investigate motor neuron disease, we recruited a very talented PhD student, Monique Bax, and she started to work on those cells. And I'll tell you a bit more about what she found shortly. But in general, our lab works on a range of different diseases, including Alzheimer's, Parkinson's, vanishing white matter disease. We've also published on brain cancer and we have some projects on epilepsy, in addition to the work we're doing on motor neuron disease. And what we try to do is use cells that are donated by patients so that we can understand disease processes and for drug discovery. And we look at the effects of how genes cause changes in those cells or how cells may work together to cause processes such as inflammation and how that affects brain function by working very specifically on certain proteins in membranes, and they're called ion channels. So the concept that we were using and still use to this day is to use patient cells to try and understand disease processes. So how that works is that a patient donates their cells. Usually this is a very accessible cell type, something like skin cells, and we can reprogram those cells into what's called a pluripotent state. So those cells then act like stem cells. And once we have those cells, we can convert them into relevant cell types, including neurons. And we take those cells, characterize them, and then test different drugs, as well as test disease mechanisms as well. So I mentioned that Monique started working in the lab in 2014, and we've been working for a number of years with Justin and Justin's lab. And this is one of the papers that is coming out of that work, also with Natalie, who you heard from earlier, and Darren Saunders, who's also been mentioned. So the paper specifically looks at protein degradation and how it's altered in cells that come from MND patients, as well as other types of cell models that carry those mutations. And it basically shows that protein degradation is altered in cells that come from MND patients. And we think that there is a big deficit in how the cells are able to deal with those proteins to remove them to prevent aggregation. So that work is still ongoing. Justin and I have published already 10 publications, but there's more in the pipeline as well. And we're continuing the work looking at MND using a number of different approaches, including machine learning, so trying to use computers to help us understand the differences between patient and control cells. And we're also using our cells to develop new therapies for MND, such as antisense oligonucleotides. And that work is also being followed up by trying to understand how to deliver these new therapies for MND as well. And that work is being done in collaboration with Cara Vine-Perro at UOW, which neatly brings me to introduce Cara for the next talk. So thank you very much, and I'll hand over to Cara now.
Thanks, Lizanne. Sorry about that delay. Look, good afternoon, everyone. It's a real honour to be here today to celebrate the remarkable achievements of my dear friend and colleague, Professor Justin Yerbury. So Justin and I have been friends for over 20 years now, and we first crossed paths as undergraduates when we were both studying science degrees here at the university. But I guess, you know, what brought us to the university to study was for very different reasons. So I was interested in pursuing a career in cancer research, while Justin was deeply driven by his personal experience and family history of MND and a drive to better understand the disease. So fast forward a few years, and after we both established our own independent labs here at UOW, we've now come together and using our complementary expertise to really develop new and innovative approaches to improve delivery of drugs to the central nervous system where they're needed. And that's what I'm going to be talking about today. Okay, so one of the biggest challenges facing the effective treatment of MND is the blood-brain barrier. So most of the talks you've heard so far are really about targeting those cellular processes. But once we've identified a target, we actually need to think about the whole person and delivering the drug to the brain and the spinal cord where it's needed to act. So the blood-brain barrier, as the name suggests, is a physical barrier between the brain's blood vessels and the cells and other compartments that make up the brain tissue. And as you can see here, and it's really essential in maintaining brain health, but it has a downside when we're trying to treat MND because the vast majority of drugs that we have developed so far don't readily cross the blood-brain barrier. So all blood vessels in the body are lined with endothelial cells, and you can see them here. So here's your blood vessel, and the endothelial cells are shown here in purple. And I want you to imagine that these people standing along here in a line are blood vessels throughout the body. And you can see here that there are gaps in between these endothelial cells, and they're there to let molecules out of the blood vessel and into the surrounding tissue. But when we look at the endothelial cells that line the brain's blood vessels, we can see that they're really wedged quite closely together.
and they form what we call tight junctions. And these tight junctions are highly selective and they only allow certain molecules through. And that means it's very challenging to deliver drugs via the bloodstream to the central nervous system where they need to act. And so a lot of the drugs, the targeted drugs that are being developed today, including the antisense oligonucleotides that Lizanne mentioned earlier, they rely on very invasive routes of administration to get them into the brain and the spinal cord. And so I guess that's how the collaboration with Justin and I started, really. It's how can we overcome this barrier to improve drug delivery? So my lab is primarily focused on developing, I guess, new drug delivery systems to improve site-specific drug delivery to cancer cells. And so this is in the context of breast and pancreatic cancer, while reducing, I guess, the toxicity to healthy tissue. And so in this way, I really need to sort of think about the barriers that we might face in the cancer context. So this might include physical, biological, and chemical barriers. And so I guess, you know, transitioning from cancer biology and drug delivery in that space to this project was not that difficult, because it's just another barrier that needs to be overcome. And so Justin and I worked together to come up with a few different project ideas. And what I'm going to be talking about today is actually some work that's been going on since about 2016. And I'm just going to highlight a couple of the exciting findings that we've got from that. So for this project, we're using a relatively new and exciting approach known as focused ultrasound. And many of you are probably familiar with diagnostic ultrasound, and focused ultrasound really is not that different. So it's a non-invasive technique that uses acoustic waves in the ultrasound spectrum to selectively and focally disrupt the blood-brain barrier. So these sound waves pass harmlessly through the bone and the tissue, and then because of the shape of the transducer, they converge on a targeted region at a focal point here. And when we inject these tiny gas-filled microbubbles into the bloodstream, they circulate through the body and they enter the blood vessels surrounding the brain. And when these microbubbles come in contact with these sound waves that we've applied through the focused ultrasound, these microbubbles start to oscillate and move around, and they impart mechanical forces on those endothelial cells, and they temporarily open those tight junctions that I spoke about earlier. And it allows the drugs to move from the bloodstream into the central nervous system. So quite nicely, what we've showed is that when we apply focused ultrasound and our lead drug formulation with to the brains of mice here, we actually disrupt that blood-brain barrier. And so what you're looking at here is some T2 weighted MRI images. And so the images on the bottom panel here are mice that have been treated with our drug, focused ultrasound for 30 seconds, and those microbubbles that I was talking about. And then the top panel are mice that have received all the treatments except the microbubbles. And so we can see negative contrast here that's indicative of blood-brain barrier opening. And we can see that that directly results in increased delivery of drug into the brain within that exact region as indicated by this yellow signal here. And we don't observe that in the control mice. So the drug can't get into the brain unless we disrupt that blood-brain barrier. And when we quantify this, we see on average that we can increase or enhance drug delivery into those targeted regions by an average of 4 times, but sometimes even higher, up to 8 or 10 times, which is very, very exciting. Importantly, all of the mice recovered when we applied this focused ultrasound. There were no behavioural changes, and the blood barrier was, we saw the blood-brain barrier was restored as early as 24 hours and fully restored 3 to 5 days later, which was fantastic. And we didn't see any signs of toxicity or neuroinflammation when we looked at the brain tissue post-study using various immunohistochemical analysis. So again, this was very exciting for us. So in order to translate these exciting findings, we next need to demonstrate that the application of focused ultrasound, microbubbles, and our lead drug formulation is actually efficacious in disease models of MND. And these are the models that Natalie, Jeremy, Kristen, and Isabel have been talking about earlier. So these mouse models that are sort of the workhorses of the Yerbury lab. we use these then to assess the effectiveness of our approach. And what we're looking for is that we can delay disease progression and prolong survival in these models. And this will probably help in the development of our lead drug formulation. But I think what's most exciting is that we can probably use this same approach and apply it to drugs that are already in preclinical or clinical development, and expand the scope of treatment options for people with MND. So with that, I would like to thank the amazing postdocs, students, collaborators who have worked on this project, and our funding partners as well. And I'd like to finish by saying that it has been a real honour and a privilege to work alongside Justin on this important project. His dedication and his passion and his tireless pursuit of a cure for MND really has been an inspiration, not just to me, but to everyone who has worked with him. And I have no doubt that his contributions will continue to have a long-lasting impact on the field for many, many years to come. So thank you so much. What I'd like to do now is to stop my screen share, if I can get my mouse back on the page. And I would like to move into a short Q&A section and take some questions from the audience. So if you do have a question for any of the panelists that are presented here today, then please add them using the Q&A function below, and I'll get through as many as I can in the next 10 or so minutes. So I do have one question here, and I think this one's probably for you, Jeremy. So maybe if everyone is happy to turn on their videos. So I think this one's for you, Jeremy. So I have a question here from Steve Oliver, who says, with the drugs, 3 may be better than 1, but did you check whether 2 was better than 3?
I'll answer that for Jeremy. So that's a good question, because minimizing the amount of drugs someone might take is important, because too much drug of any type can be toxic. We did consider it. In fact, we published a paper on 2 drugs alone, copratius and epsilon, in 2022. And that was really the starting point for the 3-drug idea. And the 2 drugs were effective in their own right against superoxide dismutase or SOD1-based MND in our cell models. However, we ended up going with 3 because the 3rd drug, telbivadine, it does add some effect to reducing inclusion formation and cell survival, but it also acts as an antiviral and could be potentially useful in the case that endogenous retroviruses are associated with motor neuron disease. But these are big level questions and quite complicated things that might take, a very long time to answer, but I hope that answer is good enough to get you started.
Thanks, Luke. Appreciate that. So I guess I mentioned earlier that a lot of the work, I guess, started at the very fundamental level. So focusing on, protein misfolding and aggregation, and you've got some really elegant models that you can use to assess, I guess, novel treatments, but also to use to better understand the disease. But has there, I guess this is open to anyone to answer, but does researching MND in this way reveal anything about other neurodegenerative diseases at all? Can anyone comment on that?
I can say something if you like. So I think the short answer is yes. So there are lots of parallels between the types of mechanisms that cause cell death in motor neuron disease, which obviously targets motor neurons, compared to other types of neurodegenerative diseases in which other neurons are vulnerable. So it's always been a big question about what causes that specific vulnerability. But as more and more research happens, we know that there are these parallel mechanisms. So something that we know, for example, protein aggregation, changes in proteostasis, those things are big factors, not just in MND, but also in Alzheimer's, Parkinson's, Huntington's, other types of neurodegenerative diseases. And I briefly mentioned inflammation. So that's another area that people are very interested in, why there are these big differences and increases and changes in inflammation that appear to be contributing to neuronal death in various diseases as well.
Excellent. Thank you, Lisanne. I've got a question here. How useful are the animal models? Do they translate well to the human system? Maybe Ron could answer that.
Yes, I can answer. Well, can we answer that? The short answer is, if you base it on previous drug trials, the answer would be no. Most of the drugs that have shown some efficacy or effects in disease models have failed to translate to treating human disease. In part, that's not because the models are necessarily bad, it's just that the models that have been developed really focus on a limited branch of motor neuron disease. So motor neuron disease has a number of triggers that causes it. And so as we move forward as a field, we need to always be testing drugs in different animal models. And Justin's work and team has been explained, it is starting to go into that direction. So as we test more mouse models and other animal models, there'll be greater success, hopefully. In addition, the big barrier at the moment too with some of the mouse models is that we often start treatments early before disease is known in those animals. However, in humans, we normally don't start treating people with motor neuron disease until they show signs of symptoms. And so another big field that would really advance and translate animal trials to humans would be to identify better biomarkers so we can start treatments as early as possible. We could possibly do that in people who we know have inherited forms of motor neuron disease, but in the majority who don't, that becomes much more difficult. So I think there's just a lot of work still to be done in that area.
Thanks, Ron. I've got a question here that maybe someone from Justin's team can answer. So eye movement seems to be somewhat immune to motor neuron disease. Do we understand what makes the movement different from other body movement?
Yes, that's a great question. And that really gets at one of the things Justin has strongly contributed to in the field, and he's even published a paper on this. Why is the eye spared so much when the rest of the motor neurons are not? And we don't know 100% sure, but one of the things that Justin has suggested and provided strong evidence for is this idea that the type and number of proteins being expressed in a motor neuron is very different between a skeletal-muscular motor neuron as compared to the one that is in your eye. So your eye motor neurons would be having a different, what we call proteome, compared to the other motor neurons in your body. And it's that this proteome difference governs how vulnerable that cell type is to developing disease. And we would think that the difference between these two cell types is an important area to look at, and it's something that Justin is actively pursuing.
Thank you, Luke. I have a question here from Ainsley. Is it helpful for current MND sufferer to volunteer for cell donation, and if so, how?
Maybe
I guess that question, is aimed at me. Thanks very much for the question. So maybe if I share my email address, we can talk about that later. But yeah, happy to talk to people later.
Thanks, Lisanne. We have another question here. Have any of the Nature Bank fractions been administered to mice with SOD1? I'm not sure if Mark's on the call and can potentially answer that question, or if anyone from the Yerbury lab's involved in that research that can answer.
I don't think they have, but Mark is definitely, he's saying he cannot join video, but I think that's on the plans for Mark, because I talked to the researcher working on that research. So they're going to test it in fish, worms, and mice at some point. They're still working on figuring out what that drug exactly is, which should happen very shortly.
Thanks, Luke. Did you want to read that response out? No, okay. So we've got a question here from Nick, and we've got a few questions that we actually can't get through, so we might just finish up on that one. So Nick is asking if there's any potential danger of opening the blood-brain barrier even for 24 hours. And I guess what I can say about that, Nick, is there's been some human clinical trials specifically where they have applied focused ultrasound to patients with MND. So not applied drug treatment at all, but just looked at the safety in the context of MND patients. And it appears to be safe. I don't know what the long-term effects may be in terms of allowing peripheral cells in, to that very privileged brain microenvironment. But the early studies tend to show that this is safe. And there's actually been a lot of work in the Alzheimer's field where they're using focused ultrasound to actually ablate these protein aggregates and amyloid fibrils to try and treat the disease that way as well. So I think there's some mounting clinical evidence to suggest that focused ultrasound is indeed safe. And certainly when you ask MND patients if they would, be up for having this type of treatment, they've done the sort of risk analysis and think that it would be worth it. But there's more research that needs to happen in that space. Look, what I might do now, unfortunately, we've got some great questions we can't get through, but there may be a way to filter them through and for us to maintain contact after this webinar. But I will have to draw this session to a close and thank all of the speakers here. And we're now going to hear about one of Justin's other projects. He's a busy man. Remarkably, Justin has written a memoir with his eyes. And after a short video, we're going to hear from Rosie Hunt from Affirm Press, who is speaking on behalf of the publishing director, Martin Hughes. So I'll just hand over for that video now. Thanks so much.
So Justin and I have been together for about 31 years. We met when we were 17, and pretty early on in our relationship, a few people in the family, in Justin's extended family, started to be diagnosed with motor neuron disease. Justin's had a steady decline since his diagnosis in 2016, and he's now 99% paralysed, but it hasn't stopped him from writing his memoir and also from continuing his important research into MND. Justin went to university to study science and went on to do a PhD and became one of the world's leading researchers in motor neuron disease research. Encouraged by Affirm Press, Justin continued to write his manuscript despite his failing health.
I am Justin Yerbury, and this is my story. I promised my mother that I would do everything I could to find a cure. I was in New York delivering a lecture when my thumb suddenly stopped working. I knew in that moment that the MND had caught up with me. I was determined to get it all down. I hope you enjoy the book and learn more about MND and the potential of research.
I heard about Justin's manuscript from a friend. She asked me to have a look at it as a favour, actually, and I wasn't expecting much, but my God, once I started reading it, I was so moved and stirred and saddened and ultimately inspired. And when I heard that Justin had written this memoir purely with eye tracking software, I just thought, what an incredible achievement. We have to publish this book. When I read the manuscript first, it was about 2/3 complete. And when I told Justin that we wanted to publish it, he became really focused, and he completed the manuscript despite a lot of health concerns. He just did a phenomenal job, and the book Fighting Fate is it's an incredible read. The circumstances under which it was written, I just cannot imagine a sharper lens on life and a greater gift than what Justin gives us with this book, which is the gift of perspective.
My name is Rosie and I'm a publicist at Affirm Press, the publisher of Justin's book, Fighting Fate. I'd like to read a speech on behalf of Martin Hughes, our publishing director, who couldn't be here today. I first read Justin Yerbury's manuscript as a favour for a friend, but I immediately became absolutely gripped by this story. Justin's story has been one of heartbreak, yes, but also determination, achievement, and love, and I knew that it would resonate with so many people. That he wrote the entire manuscript by using eye tracking software illustrated his dedication to telling his story and shining a light on this terrible disease, and I knew right away that we had to publish this book. I have published hundreds of books over the course of my career, but every so often one comes along that leaves a mark on me and reminds me why I got into this business in the first place. Can you think of a more compelling reason to write a book, or imagine a sharper lens on life and hope than this? It's fascinating, it's sad, it's inspiring, but ultimately it is a gift of perspective. I know the book has already had a profound impact on many of our staff who have come to know Justin and his story over the past 6 months. I am sure it will have the same effect on many readers for years to come. Everyone here today will already know that Justin is a hugely talented and determined scientist. But as you will see when you read Fighting Fate, he is also an incredible writer and storyteller. He brings the reader along with him on every page, starting with his idyllic childhood in Wollongong, through his schooling years and pursuit of a career in basketball, to the moment MND came into his family's life and the immense loss and challenges Justin has faced since. We also get a strong sense from the book of the beautiful relationships in Justin's life, including, of course, with his wife, Rachel, and their 2 daughters. There are so many moments in Fighting Fate I could point to that have stayed with me, but one aspect of Justin's story that stands out is his determination to pursue research into cures and therapies for MND, even as his own health has deteriorated. Starting with very little background in science and going on to achieve what he has is truly remarkable. You could easily forgive someone in Justin's position for, at a certain point, focusing only on themselves and what they need. But reading his memoir, we learn that that is not Justin. His commitment to research never wavers, and I was moved by the fact that he has advocated against discriminatory grant assessment practices, making the process fairer for everyone. I know his story will inspire readers from within and outside of the research community to do what they can to contribute to the cause of finding a cure. It's for that reason too that we are pleased to be able to donate all proceeds from sales of the book to Fight MND, a foundation that does so much for the cause. Thank you, Justin, for sharing your story with us. It's a privilege to be your publisher, and the whole team at Affirm Press is honoured to be working with you. Thank you also to Rachel for being an integral part of the process and for everything you've done throughout the process of putting the book together. I now want to introduce a reading from Justin, who has recorded a chapter from Fighting Fate. This reading comes from Chapter 4, A Strange Relationship with Death, and takes place in 1996. At this point, Justin's uncle Ken had passed away with MND, but his family have not yet learned that they carry a rare genetic form of the disease. The chapter opens with Justin sitting with his 21-year-old cousin Ashley, who has just been diagnosed with MND. We will now hear Justin read this excerpt.
I sit opposite my cousin Ashley at our small dining room table. I have trouble meeting his eyes, and my gaze drifts to the glass tabletop where Ashley's image is reflected, like his whole world, upside down. I haven't heard much of what he has said. His voice wavers and is a scratchy version of its previous smooth tones. But that is not why I don't hear him. Ashley has come to tell me that he is dying. He has a Mandy. I heard very little after these words. I had been told this tragic news before this visit by my aunt, but it hasn't made this any easier. Ashley is 21 years old, still a kid. My gaze drifts back to Ashley but doesn't dwell long before it wanders to the ceiling to rest on the repeating patterns of the decorative cornices of the half house that we were renting. What is a Mandy? I thought. How does a person die from it? It has been just over a year since Kenny had died, but I never thought to ask how. I wonder if Ashley knows more about what is in store for him, but I am too scared to ask. I had read that MND strikes people in their 40s or 50s. So it seems like a cruel nightmare that Ashley has been given this death sentence at such a young age. He has always been thin, perhaps wiry is a better word, but as he sits across from me now, labouring for every breath, he is thinner than usual. Despite everything, he still manages a smile. Ashley has always been the archetypal little brother, which has given him a chip on his shoulder. Regardless of his small frame, he never backed down from a fight, no matter the odds. He would rather go into a fight knowing that he would lose than back down to anyone. But I get a sense, as I sit across from him now, that he had lost the fighting spirit and that this foe was too big even for Ashley. Ash has a long, freckled face with a contagious smile so constant that it might have been stuck that way when the wind changed, as our parents used to say would happen. In contrast to his older brother Jamie, he has a light complexion, and his mop of blonde hair is thinning even at this young age. Ashley is my cousin, my mother's sister Catherine's second son. I grew up with Ashley and his older brother Jamie. They lived with our grandparents on Telegraph Road throughout their entire teenage years, and we would always hang out whenever we visited Young, which was most summers. While we used to spend day after day together in Young, at this moment, as he tells me he is dying, I don't know what to say to Ashley. I have no experience talking to someone who is dying. I had no reference point. Nothing I could anchor my conversation on. Because of the late death of my older sister, death has been a taboo subject all my life, and I carry that with me still. Even while death sits across from me and slaps me in the face, I still cannot acknowledge it. I realize that this is a selfish way of approaching the situation, but I cannot bring myself to talk about it. Any questions or words of comfort are caught up in the tight ball in my chest. I should ask how he really is. I should ask what he thinks about death, and I should tell him I love him. But I don't. Even today, I am unsure what frightened me the most about Ashley's diagnosis. I didn't know enough about MND, so I didn't know if he would be in pain or how the disease would end up killing him, and that scared me. The unknown, so many unknowns, make me feel uneasy. I struggled with the concept of death. An infinite amount of time of nothingness seemed the likely result of death, and I found thoughts of infinitely and death extremely unsettling. I had always felt that death was something not to be talked about. Even as a child, when I wanted to know about death and what happened to my sister Kylie, I was too afraid to ask. I didn't want to make my parents have to think about the loss of their child. Life seemed extremely fragile and such a statistical anomaly against the backdrop of the vast lifeless universe. I tried to keep these thoughts away from the front of my mind because too much thinking about it could interfere with and bog down the inner workings of my mind. I can't quite bring myself to believe that Ashley's really dying. I find myself thinking, are we sure that this disease that Ashley has is the same thing that Kenny had? Maybe they got it wrong. Kenny started with symptoms in his shoulder while Ashley first had symptoms in his throat. He lost his voice on a fishing trip, so it seems altogether different. For many months he thought that it was a typical, albeit stubborn, sore throat. He saw doctor after doctor, but nothing helped. It was only when my Aunt Gay felt that it could be MND that Ashley went to see the neurologist. I am not sure how she picked it up, but I suspect she saw something in Ashley that she had seen in Kenny. I want to ask how long the doctor said he would live, but I don't. I know that the doctors told him that there is nothing to be done and that he should get his affairs in order. I just can't bring myself to talk to him about it. I found myself feeling so helpless talking to Ashley. I wanted to be able to say that doctors knew what caused MND and that there was a treatment on the horizon. But I can't say anything. I didn't know anything and didn't understand the things I read on the internet when I tried to find answers. I don't know what made me think that I would be able to understand it. I just knew that the doctors had told Ashley to go home and get his affairs in order. And I felt that it was not good enough and that someone should be doing something about this.
Thank you all for coming. As you all know, Justin has seen it as his life's purpose to fight against the cruel hand of fate that he and his family have been delivered. And he's battled at every turn to research motor neuron disease in the hope of shedding light on its dark corners. In doing so, he has shone the light of hope for our family members and also for all people living with MND. Since his diagnosis, Justin has also fought against disadvantage and illuminated ableism as he's lived his life to the fullest and continued his research. Justin's courage to overcome obstacles, to continue working, to write his memoir, to stay with our family, and simply to keep living have been victories of mammoth proportions. So thank you all for coming to support and recognise Justin today. I'd like to offer gratitude to Trish and all her team for instigating this event and making today happen. This recognition and celebration means so much to Justin and to our whole family, as does the support that UOW has offered Justin over the years, which has enabled him to live and work with purpose and dignity. We're so grateful to Justin's devoted lab team, who, as you've seen today, go above and beyond to keep the ball rolling on MND research. Thank you to Justin's colleagues, our family and friends, and our support staff. We hope that today you have been inspired to see what can be achieved when you combine a true fighting spirit with a supportive and loving community. Thank you all.
Thank you so much, Rachel, and what an inspiring afternoon. On behalf of all of our colleagues here at the University of Wollongong, we want to acknowledge Professor Justin Yerbury's extraordinary contribution to research, his advocacy on behalf of people with a disability, and the mentorship of his team and his friendship. We've heard this afternoon how Justin and his team's research has driven fundamental new understandings of MND, and the collaboration across groups has been truly remarkable. This afternoon, I'm honoured to award the Vice-Chancellor's initial Extraordinary Luminaries Award to Justin in recognition of his significant dedication and contributions to research into motor neurone disease and the enormous advancements his research has produced. Rachel will present the award to Justin on behalf. I'd also like to thank all of my colleagues across the University of Wollongong and across the globe for being here today for this really important and impactful event. In particular, I would like to thank Justin's colleagues from the Yerbury Lab, and also to Jill McGahn, David Currow and team for their organisation of this Luminaries event. Thank you also to our audience. This event will be recorded, and also we will be able to send you a link to pre-order a copy of Fighting Fate. Again, my sincere thanks, and for everyone for participating this afternoon. Kudos, Justin, you truly are a legend. We're exceptionally proud of you here at the University of Wollongong, and we send our best wishes and blessings to you and your family. Thank you.